point, you should be able to recognize and describe the structure of DNA. You should know what it's made of, so you should be able to define what a nucleotide is. Of course, we can identify that here in a picture. A nucleotide has a sugar, a phosphate, and a nitrogen base. So this would be one single nucleotide. We know that DNA is two polymers. So here's one polymer. And so here we have 4 nucleotides. They'd be bonded together by a phosphodiester bond. So we're able to identify the phosphodiester bond here in the sugar phosphate backbone. This is a strong bond. But the 2 strands are held together by hydrogen bonds, produced by the polarity of the oxygen and the nitrogen that's actually here in the nitrogenous or nitrogen base. And we see a couple of patterns. We see the pattern that thymine always base pairs or is complementary. So make sure that you feel comfortable using that word complementary or complement. So the complement to A is always T, G is always C. This diagram also has very nicely labeled that you always have a double-ringed purine with a single-ringed pyrimidine. One of the important implications that we haven't talked about yet is that this actually gives DNA a very uniform diameter. And this will be important later when we talk about replication. That's an important characteristic, that it's always a specific width. And the final important part being the idea of anti-parallel. And so this diagram is also very nicely labeled because it shows you 1, 2, 3, 4, the 5 sugars. So when we talk about anti-parallel, what we mean is that the polymers are actually facing opposite or running in opposite directions. And the way that they give a frame of reference is they look at where the carbons are in the sugar. This 5th carbon that's hanging off the end here, we call that the 5' end. Well, each sugar has a 5' and a 3', but at the top of this strand is the 5', and at the bottom of this strand, here, this carbon at this joint here in the hexagon, this carbon is the 3' end. And so the other side is running in opposite directions, so that the nitrogen bases are both on the inside. And so whatever side is 5', its complement will be at 3'. So this is the 5' to 3' end, this is the 3' to 5' end. And so we do say that they are anti-parallel. And that is a tremendous part of the double helix structure, is that we have the they're complementary, and that they're also anti-parallel. Another component is, of course, the strong backbone on the inside and the nitrogen bases on the inside, with these hydrogen bonds being weak individually, but strong as a whole. So now that we have had a little review of the structure, which hopefully when you look at a diagram like this, you can pick out those components. The next puzzle to solve was, well, how does DNA do its job? How does the structure relate to the function? Because in science, it's all about how does it work? And the shape of it is usually closely tied to how it actually works. So the job, if we look at the job, the main component is that DNA holds the code to build any number of proteins that might be needed by any cell at any given time. So especially when you look at a multicellular organism like humans or other animals or plants or fungi or some protists, what we'll see is that those cells may actually use different amounts or sections of the DNA, but the DNA should hold the entire genome or genetic code for the organism. And so we're going to be able to build proteins. Of course, proteins are going to give us traits. Some proteins are pigments. They are going to absorb and reflect light. So this, for example, gives you eye color and skin color. Some proteins are going to be enzymes. So they're going to control the rate of reactions. Some proteins are going to be structural components, like muscle. And so DNA holds the code to give us proteins, which give us our traits. And we're going to talk later about transcription and translation and the actual process. Keep in mind, we're not actually changing DNA into RNA. We'll be doing what I call rewriting or base pairing to get RNA. And then we use this code to actually assemble the amino acids. So this is where amino acids actually get assembled. So this is a little bit of review maybe from your introductory bio class, preview into what we're going to be talking about later. But another really important part of the job is the concept of replication. So it needs to be in any cell. And so if we look at a multicellular organism that's going from one cell to two cells, for example in mitosis, or if we're preparing for sexual reproduction and we need our offspring to have DNA, we know that the first step before cell division is actually replication, that the DNA gets copied. So one of the mysteries that needed to be solved is how does DNA get, and I'm going to put it in quotes, copied? How do we get the DNA into every cell? So there were two gentlemen, and I'm sure a whole host of lab assistants who helped out, Meselson and Stahl at the California Institute of Technology, and they designed an experiment to test how DNA actually replicates. And again, they used isotopes. So we want to make sure we really know what an isotope is. It has a different, so it differs in the number of neutrons. And so what they were using was heavy and light nitrogen, like in the nitrogen bases. Now these days you could just order yourself some of this. But at that point what they did is they grew their DNA. So this original DNA was grown on a specific type of nucleotide, and they were always grown first on the normal, they were grown first on the heavy nitrogen. So they grew them on nitrogen 15. Nitrogen normally only has 14 neutrons, and they grew it on heavy. So that generation after generation after generation after generation all have this heavy nitrogen in the nitrogenous bases. And so what they were looking at, and there are really good diagrams usually in your textbook that go along with this as well, what they did is they took the cells that had uniform heavy nitrogen and they allowed them to divide but the nutrients that were provided, where are those nucleotides going to come from? I'm going to make more DNA, I have to have more nucleotides. Those nucleotides come from your nutrition. The nutrients that were provided were only light nitrogen, or N14. So, they grew it on heavy nitrogen. All the nucleotides in here for many generations are going to be a specific type. They're going to be heavy. And they were actually heavy. The chemical they separated it in, these dropped more to the bottom. Then they allowed it to divide once on a medium or food source that had only light nitrogen. And that's what we're seeing here in the blue. The light nitrogen is representing the blue, the yellow representing the heavy. So Meselson and Stahl said, well, there are probably three likely outcomes. The first suggested outcome is called conservative replication. The idea being that it unzips, we copy it, and this one rezips, and here's our completely new one, and that's called conservative. Dispersive replication is the idea that somehow the DNA broke up into pieces and recombined so that the two were just a mix of the parent, that you're getting a mixture of the parent and the new. And the third is called semi-conservative replication, and this theory was that as we unzipped, we added new nucleotides, and so the two resulting were actually going to have one original from the parent and one newly constructed strand. So what did we see? The results of Meselson and Stahl. And this is actually an experiment that the AP curriculum wants you to know. The results are that after the first round of division, so this would be like our F1 generation, they saw that, so what this looked like in the test tube was that there was one band of heavy, that all of the DNA in the cells were heavy. After the first round, what they saw was whatever it was all medium. It's not the heavy form anymore, that it's all somewhere in the middle. There weren't any DNA strands down here that were light or heavy, so we didn't have a separation of light versus heavy. So somehow the DNA strands were a mixture of the two. So this first round, the F1, actually proved that conservative replication is not what was happening. We didn't have some heavy and some light. Everything was medium. So now we're down to these two. So how do we prove which one it is of those two? So they did a second round. And this would be, second round of division would be like the F2 generation. And if this happened again, what we should see is, again, some hybrids. Everything being equal, that basically you're cutting it up and dividing it equally. What they saw is that there were some that were medium, and then keep in mind, now we're growing it on the light, so guess what? They saw some that were all light. Because after the second round, and this is what we actually saw, after the second round, remember we're growing it on blue. So after the second round, what they saw is that this strand split so that I had a blue as a template and a yellow as a template. Over here I'd have a yellow and I'd have a blue. We're growing it on this blue, this heavy nitrogen, and so every time I add the blue, and this is actually what they saw in their data, that 2 were of the medium and 2 were of the light. And I didn't do a good job with the scales on my diagrams, but they're actually really good ones usually in your textbook as well. But the key concept is that if this had happened, we'd have bits and pieces so that all of the resulting DNA that was made would be equal amounts of old and new. And here, because this is splitting, we see that the remnants of the heavy nitrogen is actually still complete. It's still in one section. It hasn't been broken up or dispersed. And so that's why it's called semi-conservative, because each strand that is produced contains a whole strand from whatever the parents were. So the big concept of Meselson and Stahl that we want to be able to leave with is that they were able to prove semi-conservative replication. Now there's a lot more to know about replication. I want to give you a little preview into that. There's a whole schematic down here that's going to show how molecularly this actually happens. So we're going to get into that next time. But the main idea is that we're going to unzip, which is breaking the hydrogen bonds. We're going to use complementary base pairs. To attract the nitrogen bases. We're going to build a sugar phosphate backbone. And the last thing is actually proofreading. We're going to see a step that an